Started. Thank you, everyone, for coming along to part 2 of Professor Stephen Sen's webinars about design analysis of N-of-1 trials. I'd like to begin by acknowledging the traditional custodians on the lands on which we meet today and pay respects to elders past, present and emerging. We extend that respect to Aboriginal and Torres Strait Islander peoples here today. So 2 weeks ago in the first session, we had Stephen presented a very interesting session, and we heard a lot of the details about fixed effect analysis and connections with meta-analysis. And we're going to be hearing more about that today, and in terms of random effects approaches, I believe, and then looking at binary data and some design issues, I believe, Stephen. But I think I'll hand over to Stephen now. So there's 2 parts to this. There's a lecture 3 and a 4, and we'll have a little 5-minute break in between the 2. Just a very brief bio of Stephen, in case you missed the first one. Stephen Sen worked as a statistician, but also an academic in various positions in Switzerland, Scotland, England, France, and Luxembourg. He was head of the Competence Centre for Methodology and Statistics at the Luxembourg Institute of Health. And he's the author of some very readable and important books that I really recommend statisticians read if you're working in clinical trials: Crossover Trials in Clinical Research and Statistical Issues in Drug Development. And the most recent edition of that was in 2021, and also has a more sort of general reading called Dicing with Death. In 2009, he was awarded the Bradford Hill Medal of the Royal Statistical Society, and 2017 he gave the Fisher Memorial Lecture. He's an honorary life member of ESI and of ISCB. So I'm very happy to have you here for part 2 of the N-of-1 trials. So I'll hand over to you, Stephen. Thank you.
Okay, thank you, Andrew. Just to say what I hope to do in this lecture, I'm going to revisit the purpose of clinical trials, just remind ourselves of what we were looking at 2 weeks ago, show how some of these questions are best answered using mixed models, and I'm going to discuss a particular mixed model that can be used for analyzing designs randomized in cycles. And then I'm going to make a link to meta-analysis. And consider the application of empirical-based reducing shrunk estimates and give some practical advice regarding analysis. And in doing this, I'm going to skip over the algebra. So a number of the slides have got some algebraic expressions on them. I'm not going to spend much time on them because I think we're just wasting time if we do that. If you're enthused to have a look at those, you can look at those in more detail, because I'm going to be providing copies of the slides to Andrew and Michael to disseminate. So here's a reminder of the possible objectives of an analysis. And the basic point here is that I think people sometimes forget there are these objectives, and they sometimes imagine that they can all be analyzed using the same analysis, or perhaps they don't imagine that, but they sort of flip between one and the other without really saying what they're looking at. So the first one is one of the treatments better? And that's the sort of thing that we analyze using the significance tests that were championed by R.A. Fisher more than 100 years ago. What can be said about the average effect in the patients that were studied? For that, we have estimates and confidence intervals. But that's for the patients actually in the trial. What can be said about the average effect in future patients is obviously a more difficult question. And also, we might be interested in trying to say something about what the effect is of a given patient in the trial. And N-of-1 trials are particularly suited for this. And what could we say about a future patient not in the trial? These are all questions we could be interested in answering using N-of-1 trials. So far, we've looked mainly at the first of these 2 questions. To paraphrase, can we prove the treatment works at all? And what can we say about how it worked for the patients that we studied? And the point here is that the population was the population of all possible randomizations. In our particular example, we had a look at 8 possible sequences that there were, and there were 12 possible patients. So the number of possible randomizations was 8 to the power of 12, and that's a very large number, as you can see. And the point being that although we studied each patient 3 times under both conditions, under each condition, we couldn't, because it's factually impossible, study them on the same occasion using the 2 conditions. And it's always possible that their resting state of illness will be different from occasion to occasion. So we don't, in fact, have a perfect counterfactual. We may think we do because we're running an N-of-1 trial, but we don't. So now we're going to look at more ambitious questions. In particular, can we provide better estimates for individual patients than just the average? What could we tell someone who'd actually gone to the bother of enrolling in one of our N-of-1 trials? Could we tell them what was best for them, not just what was best on average? And if patients do vary in their response to treatment, what can we say about the effect of treatment in general? So not just for the patient studied, but maybe for patients more generally. So this means that our horizon changes. We no longer think purely of the causal questions as to what really happened in this trial, fixing the patients, but we now think of the patients themselves as being in some sense representative of other patients. So there's a warning here that this involves strong assumptions, and I think this is often ignored. We tend to sort of assume that clinical trials are in some way involved in random sampling, but that's not the case. Any statistician who worked in survey work would be horrified at this particular idea. And we sometimes then say, well, okay, we know that's not true, but we think that the patients are representative in some way of the target population. But that itself is requires extremely strong assumptions, because the target population, of course, lies in the future. We study patients now, but we want to treat patients in the future, and things change over time. You only have to think of the way in which new variants have arisen in the COVID pandemic to see that this is the case. So the warning here is that one can try and answer these questions, one can try and address the uncertainty involved in talking about future patients, but it's only part of the story. There's only so much that statistics can do. Assumptions are required as well. And I'm now going to have a look at a model for this. So the model is as follows. I'm going to consider that we have an outcome Y, or in occasion S equals 1 to 2, of cycle R, I equals 1, 2 to K. In our particular example, we had 3 cycles, so K was 3. For patient I equals 1 to N, where in our particular case, N was 12. And we're going to need a number of different error terms here. We've got the within cycle, within patient error, which is epsilon IRS. The difference between patients as a whole, which is, between cycles, which is beta IR. And then between patients, lambda I, we imagine that different patients have different degrees of disability or illness or sickness or whatever. So different patients given the same treatment, it will in the long term have different levels of outcome. And then the new thing here, the thing which is really important if we're going to have a look at random effects, is we have a TOR sub I. We have a treatment effect that varies. So we're now supposing that the treatment itself might vary depending upon which particular patient we're looking at. And each of these has got an expectation. For the first two, I'm going to assume that there's 0. For this one here, for this one here, there's some sort of average effect lambda for the patients. And then we're going to have a look at this particular term, TOR, capital TOR in particular, which is the average effect for the patients studied in the trial or studied as a whole. And that has a particular variance associated with it, which I'm using the symbol psi for. Now, the whole point about this is we need to be a bit careful with the indicator, because the treatment that a patient is given on a given occasion for a given cycle will vary depending upon the randomization scheme chosen. And this means there is a particular difficulty in indexing all of this. And one way to deal with this is to have a sort of treatment indicator, ZIRS, which is given the value half or minus half, depending upon whether the treatment allocated to that patient in that cycle is A or B. I'm going to assume all stochastic terms are independent of each other. And we can now make our focus one of two things. I can either look at the actual average of those TOR-I terms. I can try and work on what they were for the patients in the trial, or I can have a look at capital TOR, which is somehow some general effect which I might apply, which I might imagine would apply for patients in general. So the first of these is effectively the fixed effect, and the second of these is the random effect. And I'd just like to make one point here about meta-analysis. It seems to me that people often get confused about meta-analysis. They think that whether you're doing a fixed effect or a random effect, meta-analysis depends on whether heterogeneity is present or not. In my view, this is quite wrong. It depends on the question. If the question is what happened to the patients in the trials that were in the meta-analysis, that's a fixed effect analysis. If the question is what might one say about patients in general in clinical trials, possibly including trials that have not yet been run, then that's the random effect meta-analysis. So if we have a look at the random terms, just to summarize this, lambda sub I is the random main effect for patient I. Beta sub IR is the random effect for cycle R within patient I. Epsilon IRS is the random error term for occasion S within cycle R within patient R. And TOR sub I is a random treatment effect for patient I. It's this thing which is particularly of interest here. Some of the terms make very little difference to the estimates produced, whichever approach is used. In effect, the first two terms can be eliminated very easily by differencing. So an analysis of differences simply gets rid of them, the differences within cycles within patients. But the random by treatment by patient interaction is handled very differently depending upon what question we're answering. If we look at the expectations and variances, this is just summarizing what I've already said. Then the first two rows here of this particular table are really unimportant. They're going to be eliminated by analysis. What's important is separating between these two things here. Sigma squared is the sort of bedrock random variability that applies the fact that patients, even when given the same treatment, will show different results from occasion to occasion because of random influences in their lives or for whatever reason. And the second is the thing which personalized medicine is particularly interested in, although in my opinion often fails to address properly formally, and that is the patient by treatment interaction term, the variance of the way in which patient results can vary, differences between the two treatments can vary from patient to patient. So these are the two important things here. If we revisit our previous estimator, the fixed effect estimator, then it was an estimator of this particular form. All we're doing is taking these within cycle differences, which is the numerator here, dividing them by something which flips between -1 and 1, depending upon which treatment was given in the second period of the cycle or the first period, and dividing by the total number of cycles that there were. If we have the same number of cycles per patient, then this simplifies to the following. The variances of the estimator that we're talking about. Well, as an estimator of the mean treatment effect for these patients, then in fact, we only need to allow for the random occasion within cycle, within patient variation. It means we can ignore the chi-squared term. Why can we ignore it? Because we're going to imagine that as we repeatedly randomize, we keep the patients the same. That means that the total effect of all of their different responses to treatment is fixed. That element of it is fixed. We're simply estimating the average effect for these particular patients. So that particular thing does not become a relevant variance. And so we're simply using then in that case, the within subject variance to estimate variability. However, that's not the case if we now regard the patients as being somehow a sample of all patients. And we could imagine that if we were to repeat the trial, we would actually repeat it with different patients. In that case, this sampling of these effects is important, and that particular variance is governed by psi squared. And so that particular variability will reduce the more patients we study, but it cannot reduce simply by increasing the number of cycles. So we now see, if we have a look at the particular, perhaps easiest to see the balance case here, if we look at the variance of this particular treatment estimate, we not only have the contribution of sigma squared divided by the total number of cycles that we studied, that's to say, patients times cycles. We can generate more information by having more cycles per patient. But we also have a contribution of the chi-squared term, and this can only be reduced by studying more patients. I should make a point about the estimator here, because we're going to change the estimator slightly in future. The estimator is only optimal in the balanced case, or if it is the case that psi squared is equal to 0. And B, whether or not this is optimal, the variance for this purpose is only the same if psi squared is equal to 0. So let's just remind ourselves of the example we've been working on. We're going to have a look at the balanced case, first of all, where basically the same estimator for the overall treatment effect applies, and then we'll have a look at the unbalanced case. Here's a representation of the balanced case, but just looking at it in terms of all of the cycles, the 36 cycles. So each cycle there, the 3 cycles in the 12 patients, is represented by a point. I don't really need to separate them by patient, because I'm assuming that I'm not interested in the differences between patients here. They don't matter. The patients are just grist to the mill. They're just a way of getting more information, or getting information. And I've done a scatter plot here in which the value under treatment B is plotted against the value under treatment A, and the black diagonal line is a line of equality, and we can see what we're already familiar with from this particular example, and that is that most of the points lie to the left and above the line of equality. So there seems to be general evidence that treatment B produces a higher forced expiratory volume in 1 second than treatment A. Treatment B, in other words, is better than treatment A. This is the same thing in the form we've seen before. This is the trellis plot. Now the results are separated by patient, which would of course enable us perhaps to see whether there are particular patients for whom the treatment B is more beneficial than A, the majority presumably, but whether there are some patients for whom the reverse is the case. This doesn't actually seem to be so from this particular scatter plot. Here there are 3 blue circles, sorry, are the individual cycle effects that we saw on the previous diagram, but now plotted patient by patient. And the red asterisk is simply the average for that particular patient. We can see here, by the way, that although some of the blue dots are below the line of equality, in actual fact, what we have is that the asterisks are always above, albeit sometimes very close to, the line of equality. So we have 2 simple analyses which we encountered before. I'm just listing them here on the next slide. Next 2 slides, I'll go through them in detail. So the match pairs T using cycles to define pair. This is an analysis in GenStat, by the way. So the variate I've called diff. There are 36 values. The mean difference is 188.7. Its variance is 25,356. And that leads a standard deviation of 159.2. And the standard error of the mean is simply this particular figure divided by the square root of 36, which is 6, and we get 26.54. The 95% confidence interval is given, and this is nicely away from 0, and so it's not surprising that the t-statistic is large, 7.11 on 35 degrees of freedom, and the p-value is less than 0.001. GenStat doesn't standardly give you p-values in greater detail than that, although you can request them, which is sort of quite amusing. The New England Journal of Medicine, I think, also has, or used to have, the policy that you can't give values in greater precision than this, or you shouldn't do. But a recent movement to try and improve the reporting of p-values suggested that p-values that were not less than 0.001 were not of interest anyway. So there's some particular difference of opinion in this in statistics. If we look at the summary measures approach, we reduce everything, first of all, to an average per patient. And that means that our sample size is now 12 and not 36. Since the results are balanced, the mean is exactly the same as it was before. So we had 188.7, which is the average of the 36 cycle differences. And we have 188.7 again. It's exactly the same average, just carried out in 2 stages. First, the 3 differences per patient are reduced to an average per patient, and then the 12 averages per patient are reduced to an overall average. And obviously, since the particular design is in balance, we're not going to get any difference here. However, the variance, the standard error of the mean will not be the same, because the variance doesn't have to be the same proceeding in this particular way, and the standard deviation doesn't anyway. In any case, the standard deviation, of course, is the standard deviation that applies for an average of 3 values rather than an average of rather than 1 single value. But it should, one would imagine, go through to the same thing as the standard error, but there's a slight contribution from the patient by treatment interaction, which is not the same depending on how you do this. And so the standard error of the mean is 28.38, whereas previously it was 26.54. The t-statistic is now, not surprisingly, slightly lower, since the standard error of the mean is higher. More important contribution perhaps here is that we only have 11 degrees of freedom. Because of the way that we proceeded, the p-value is still less than 0.001. So the previous approaches were 2-stage approaches. The first stage was to reduce the data to meaningful contrasts, either mean difference per cycle or mean difference per patient. The second stage is to analyze the contrast using a matched pairs T, based on 36 within cycle differences or based on 12 within patient mean differences. An alternative is to use a mixed model. In doing this, I want to be quite clear that there's nothing magic about a mixed model. A mixed model is, from one point of view, not radically different from a 2-stage analysis. It will often produce very, very similar results, and advice I often give clients when I'm consulting who want to have a look and understand the mixed models is to try a summary measures approach and see how similar the results are, and frequently are very similar. The difference really for an overall analysis arises in the unbalanced case. But mixed models are more powerful for certain purposes, and that's why we love them. They are particularly good at having a look at individual random effects. But provided there are no incomplete cycles, the following 2 approaches are equivalent, by the way: a model for the whole data or a model for the differences. So R code, this is the R code for approach number 1. I'm sure many of you are much more competent in modeling with R than I am. I'm using the LME4 package, which I think is the Pinero and Bates package here for mixed models. And I'm simply encoding that particular formula that we had several slides ago. This one here. Separating out those things which are random and those things which are fixed. The random stuff comes here, essentially the random interaction I'm looking at. And then you will see that what I get is I hope, the same standard error I got before using the summary measures approach. 28.38 we have there. And the t-value here is 6.65. That's the summary measures approach. And using the mixed model approach, 6.65 also. The estimate, of course, is the same, but that's hardly surprising. So the point here is that the mixed model approach and the summary measures approach give us exactly the same answer. Approach number 2, this is using differences. Here I'm simply modeling the differences. And then in that case, I've actually reduced them to differences, I think, of B minus A, treatment B minus A, and I can simply model them in this particular way. And we get exactly the same answer we had before. Makes no difference. Approach number 3, this is the summary measures approach just for reference. Here I'm, what I previously did in GenStat, I'm doing here in R. And that gives us 6.64 0.49, so we get the same, essentially the same t-value that we had before. Now I can also look at this in terms of a meta-analysis, and this is what I've done here. This is a forest plot of random and fixed effects meta-analysis in GenStat. A point to note here, by the way, is that confidence intervals are all of the same width. This is because what I have done is I've used the pooled variance to estimate the variance per patient. I'm not allowing each patient to provide me a separate estimate of the variance. I'm allowing them to provide a separate estimate of the mean, but not of the variance. And this is because the degrees of freedom which are available per patient are too few to reliably estimate the variance per patient. If I estimate the variance per patient, I'll get very different answers from one patient to another. And in a meta-analysis, I will end up weighting the results very unequally, which will be far from optimal. So I'm imposing the assumption that the variances are all identical. And the fixed effect meta-analysis and the random effect meta-analysis only differ slightly. It's a bit difficult to see, but you can, the point estimate is the same anyway, because we've got a balance case. But you can see that the confidence interval is just slightly wider for the random effect meta-analysis. Using the meta package in R, this is Schwarz's meta-analysis package in R, which some of you may be familiar with. You've got there the fixed effect and the random effect model results at the bottom. Again, I've used the same standard error for all of the patients. So I've allowed the treatment estimate to vary patient by patient, but the standard error, 88.85 in every particular case. And since there are 12 patients and the standard error is the same for each patient, then in that case, the weighting will be exactly the same for each patient. And of course, since it's a balanced case, the weighting will be the same for the random effect analysis as well. So the weighting does not differ between fixed and random effects analysis. What differs is the interpretation of the variance, and you can see if you have a look at the bottom that the confidence intervals are slightly wider. For the random effects meta-analysis, the lower limit is 133.1 rather than 138.45, and the upper limit is 244.35 rather than 239.00. So what can we conclude from this? Several different approaches to analysis give exactly the same estimate and standard error of the estimate. And these are the three approaches I've used: the mixed model approach, the summary measures approach, and the random effects meta-analysis approach. So the question is why? Why do they give the same answer? Well, what the mixed model approach does is it estimates the two components of variation. The two components of variation here are the patient by treatment interaction and the pure within patient variance. These two components are estimated separately, but they're then added together for the purpose of calculating the variance of the treatment estimate. The summary measures approach sweeps everything together and it estimates the variance using the combined effect at the patient level. So effectively what it does is it estimates the same total variance, but it doesn't separate them. So the mixed model approach, the summary measures approach, give the same answer. The former has the advantage that it can separate the variance components, which is useful for different purposes, but it's not actually relevant for the purpose here of estimating the mean in the balance case. The random effects meta-analysis actually starts by using the wrong variance. It starts by saying, oh, well, let's have a look and see what happens if these variances are correct, and says, well, wait a minute, actually, the results are varying from patient to patient slightly more than will be indicated by using the within patient variance. So let's inflate the overall variance by this particular term. So we then arrive at exactly the same situation we get with the other three. Now, the reason I'm stressing this is there's a lot of software around for doing random effects meta-analysis, a huge amount. There's been a really terrific development in this particular field, rather less development in the terms of software for N-of-1 trials. But by tweaking the results from N-of-1 trials, just a little tweak to calculate the variance, you can leverage the random effect meta-analysis. And this means you have a lot of powerful routines available for doing all sorts of interesting things with N-of-1 trials. So just to summarize the issue of summary measures versus mixed model, the summary measures approach is a simple valid analysis if you wish to test the null hypothesis that the treatment effect is 0. It assesses the total contribution of the various variance terms. It's fine and indeed robust for various purposes. But for others, you need to separate the variance components, and that's what the mixed model approach allows. And I'm now going to show you what use you can make of these. And this is using a technique called empirical Bayes. So I'm going to suppose we've carried out a long series of N-of-1 trials. And we have estimated the following: the average treatment effect TOR. And we've also estimated the variance of the true treatment effect from patient to patient. This is what we've called psi-squared in our model. Now we encounter a new patient. We want to tell this patient something about what their long-term expectation is if they take treatment B rather than treatment A, the expectation of the difference it will make to them, and also how certain we are about this. Well, if we don't study them, if we just say, you know, I don't propose to enter you into a trial, I'm just going to give you what my best hunch is, and I can tell you that the average effect of B minus A is positive, and this is the result TOR, and the patient then says, yeah, but how likely would it work for me? Then we can actually quote psi-squared to them. Obviously, it's going to be a bit difficult in this particular physician-patient conversation, but we can try and make some way of describing what the variation of the effect is. Psi-squared is the variation of the long-term effect from patient to patient. But suppose instead the patient persuades us that we actually study them also in an N-of-1 trial. You did some N-of-1 trials before, why can't you do one for me? So what we can do now is we can estimate the treatment effect for that particular patient, let's call it TOR hat, and we can estimate it in K cycles, and it will have a variance of 2 sigma squared over K. We're completely ignoring the result we've got from all other patients. Now, the interesting thing here is we're not necessarily doing better for that patient by just using their own results. It really depends on how often we've studied them and how important the within-patient variability is. But we can have the best of both worlds. We can use both estimates. We can use the global estimate and we can use the local estimate. And effectively, we're using the global estimate as a sort of, and its uncertainty as a prior distribution for the current estimate. And this is why it's referred to as an empirical Bayes approach. So if we look at this in more detail, we use the weighting formula, which is the same weighting formula which underlies Bayesian analysis. It's the same weighting formula which underlies meta-analysis. And it works as follows. We look at things in terms of precision. Precision is the reciprocal of the variance, and the posterior precision is equal to the prior precision plus the data precision. The prior precision here is the precision, the reciprocal of the variance of the result we got for all patients, the large number of patients we studied previously, and the data precision is the precision for this particular patient. And the posterior mean is the prior precision times the prior mean plus the data precision times the data mean divided by the posterior precision. These two particular formulae, by the way, which were known as long ago, at least as the 19th century, Airy's book of 1850, about thereabouts, uses this particular formula, not for N-of-1 trials, but for astronomical observations, has been around a long time in statistics. The posterior variance is simply the reciprocal of posterior precision. I'm going to skip the derivation here of the formulae in MathCAD. You can have a look at that if you like. And just give you here this particular slide, which I'm also not going to go in detail. This is a list of various formulae that may be important. The first one is the standard fixed effect meta-analysis variance. The second one is the variance of the shrinkage estimate. It's the variance of the thing I just described. The third one is the random effect variance estimated for the overall mean. We've already encountered that. And the last one is the variance of the shrinkage estimated for a future patient on the assumption that the necessary parameters have been well estimated. It's the asymptotic version of the second formula above as n approaches infinity. So here's an example of meta-analysis. Here I'm having to use a number of different meta-analysis packages. For example, as far as I No, Guido Schwarz's excellent meta package doesn't actually include shrinkage estimators, but Wolfgang Fischbauer's metaphor package does, and I've forgotten why I've included the R-meta package as well. But anyway, here are some various packages that we've loaded, and we can use, we can set some meta-analysis methods. And here I've carried out random effects meta-analysis using the metaphor package, and we've got the particular results at the bottom that we had before. And the important thing here, this is the bit I wanted to get to, is what it does is it actually produces some so-called shrunk estimates for us. So it starts with the estimate, the data estimate for that particular patient and the standard error. And then from that, it shrinks these values towards the overall mean. So you'll see, I've got a plot of the thing here. If I take the naive estimate, which is the results just from the individual patient, plotted on the x-axis, and the shrunk estimate on the y-axis, if there were no shrinkage, all of the values would lie along the black diagonal line, which is a line of equality. Instead, what you find is the blue points are shrunk closer towards the overall mean, which is 188.7. So I'm mixing the two. What I'm doing is I'm mixing the local estimate for that particular patient and the global estimate to get these particular shrunk estimates. So not only do I modify my estimate, but actually I'm also able to reduce my standard errors. You can see there's a considerable reduction in the uncertainty by being able to use these two estimates. Okay, so far we were considering the balance case, and that's interesting to get our ideas fixed, to illustrate that from one point of view, these methods are very similar. But now I'm going to consider an unbalanced case, and the previous example is going to be modified to have some imbalance. And I'm going to assume that patient number 11 dropped out after having been treated in 2 cycles, and patient number 12 dropped out having been treated in 1 cycle only. Now, some complications result, but approaches that were previously equivalent no longer are so. And the mixed model approach is slightly more efficient than the summary measures approach, because the summary measures approach will do the following. Once you've reduced everything to an average per patient, the computer doesn't know that these averages have different information with them. And all it will do is it will weight them equally. But they're not measured with equal precision, and that's something that we can use. If we have the data per patient estimate here, then in that case, the standard errors will no longer be the same. The standard errors will be the same for all the patients with the same number of cycles because they're based on a pooled variance. But patient number 11 only has 2 cycles, so the divisor is square root of 2 rather than square root of 3, as would be the case for producing these standard errors. And patient number 12 has only got 1 cycle, so their particular standard error is the same as standard deviation because I'm only dividing by the square root of 1. This is a particular trellis plot. That's the trellis plot in GenStat, by the way. This is the trellis plot in R. This is patient number 11 with 2 cycles data only, and this is patient number 12 with 1 cycle only. Here I've got in GenStat, I've also got the averages, and you can see that the average for patient number 12 is the same as the 1 cycle value, which is obvious since there is only 1 cycle to average. So if I do a model on within cycle differences, then in that case, I get an estimate for the random term, the patient error term, and for the residual as follows. And when I come to calculate the shrunk estimates, this is what GenStat tells me. The value for patient number 12 is shrunk much more radically, and for patient number 11, somewhat more radically than for the other patients. The other patients lie along the same line of shrinkage because they have the same amount of information locally from their own results, whereas 11 and 12 have less information, and therefore they are shrunk more radically to the overall mean. which is now somewhat different because of the unbalanced case and because some data are missing. So this is the result of doing that particular analysis, and the standard errors that I get here. Interesting thing is that it's not so radically different for patient number 12 and 11 because I'm able to leverage the overall information that there is. This is various analyses in R. I'm not going to go into. You can have a look at this yourself. And this is the output in R. This is the original data, sorry, here. I don't have the output for some reason. They've got the output as a graphic, that's all, which I hope is identical to the GenStat one. These are some further variance formulae that I'm not going to go into. We're coming up to the 5-minute break now, and I'll just give you some conclusions here. The important point here is that with the help of mixed models and empirical Bayes, then shrinkage approaches can produce better estimates. But modeling all this correctly can be tricky, and having alternative approaches and even alternative software to check can be helpful. I often find it useful to try and do things using different packages, and we'll see in the next lecture this is particularly useful where in actual fact the different packages even give slightly different results, and that's perhaps a salutary lesson. One thing I should say is it's also there are practical issues here which are important, which I've not addressed. If you want to use shrinkage estimates for particular patients who are in a set of N-of-1 trials, then obviously these shrinkage estimates are not available for the first patients because you haven't estimated results for patients as a whole. They're only really available for later patients, or you could possibly, in theory, revisit your recommendations for the earlier patients. But one has to appreciate that a lot of this, all of this, in fact, and this is true of all clinical trials, takes place in real time, and one's ability to use the results is constrained by this particular fact. Okay, we'll now have a break for 5 minutes, and then we'll resume with lecture number 4. Thank you.
Okay, thanks very much, Stephen. So I've asked for questions in chat. We don't have any yet, so we'll just take the 5-minute break and we'll have the sort of consolidated questions at the end. So it's 5:43 Melbourne time. So let's take a break for 5 minutes, come back 5:48. 5-minute break. Okay, thanks very much, Stephen. Yes.
Okay. So this lecture is in 2 parts. In part 1, I'm going to have a look at approaches to modeling binary outcomes. And in part 2, I shall have a look at some matters to do with planning. So as regards the binary outcome work, then I have some people to thank. One is my former PhD student, Boikanyo Makabati, who's now back in Botswana. And he was funded by the British Council and by Boehringer Ingelheim. And some of this work was carried out during the time I was on sabbatical at NSAI, which is one of the French grandes écoles based in Rennes, and also as a result of work with the IDEAL project, which was looking at rare diseases. So there are 3 kinds of binary outcome, in my opinion. Rather rare is the case that the outcome that is measured on a given occasion is itself binary. People often talk about binary outcomes in connection with clinical trials, but very often, very few things rather are binary. Sort of thing I can think of perhaps is where you're actually trying to deal with a particular infection, and the infection is either completely resolved or not. The original outcome is continuous and is dichotomized. Most binary outcomes are actually of this particular sort. And the dichotomy can either be in space or in time or in both. So for example, you can have a look and see whether a particular patient recovered a stroke to have a particular level on the disability score at a particular time. The third sort is the original outcomes on occasions within a cycle. This applies to N-of-1 trials, have been reduced to a preference by noting which of 2 treatments, A or B, gave the better response. So if the original outcome is continuous, the preference is a binary measure. In other circumstances, you can have a tie and one form of analysis to condition on a preference having been expressed. So of these three types of binary outcome, I'm going to have a look at the third and then the first. I'm not going to have a look at the second. It was assumed in the previous slide that where a preference was expressed, it was expressed for each cycle, but of course you could do the same per patient. So the former is analogous to the analysis of differences within cycles that we consider for continuous data, match pairs T, a mixed model, and the latter is analogous to the summary measures approach. Here's an example. This is a sort of standard guinea pig of clinical trial use. This is from a fairly early paper by Roman Yeshka, Gordon Guyatt, a famous name associated with evidence-based medicine with this. And this was clinical usefulness of amitriptyline in fibromyalgia. And this is the results of 23 N-of-1 randomized controlled trials, by which they mean that 23 patients were studied using the sort of N-of-1 trial protocol that we've looked at so far. This is a possible representation of the results. Here, what you have on the left-hand side is a sort of dot histogram showing for each of the 23 patients. So patients are spread out along the x-axis according to patient number. And patients were treated on 3 to 6 occasions. So each patient has got 3 dots, 3 vertical dots associated with them corresponding to occasion 1, 2, and 3. And here I have color-coded them with a filled blue circle indicating that amitriptyline was better and an open red circle indicating that placebo was better. So this is a summary of all the results, and you can see that what I've done, I think, here is I've sorted the results for each patient to make it easier to see, so that the amitriptyline better results are at the bottom and the placebo better results are at the top. So occasion here has been sorted according to response. It's not actually in time, I don't think. If you want the actual numbers, the numbers are on the right-hand side. And so for a given patient, you can record the proportion of occasions on which they preferred amitriptyline. And so for patient number 1, who has 3 open red circles, then in that case, you can see on none of the 3 occasions was amitriptyline preferred to placebo. So the value of P there for that patient is 0.00. On the other hand, patients 18 to 18, 20, 21, 22, 23, 19, and 14 all had preferred amitriptyline on every occasion. These were studied on 3 cycles in some cases and 4 cycles in others. And so these are basically the proportions that you have. But what's also important, in addition to the proportions, I suppose, is the number of occasions which are studied. You could, in theory, have had a proportion of 0.5, although this was never observed here, in which you'd had 2 out of 4 or 3 out of 6 preferences for amitriptyline. So if I summarize it in terms of cycles only, if I go directly to cycles, ignoring the difference in patients, it's analogous to what we did before, then I could carry out a one-sample binomial test. And this is what I've done here. The analysis in GenStat tells me that the overall proportion is 0.675. That's simply the result, the average of all the cycles. And if I want to test the null hypothesis that the proportion is equal to 0.5, why 0.5? Well, 0.5 is indifference between placebo and amitriptyline. Then in that case, the p-value is 0.003. And the 95% exact confidence interval proportion is from 0.56 to 0.78. On the other hand, what I could do is I could reduce it to a preference per patient. So if you have a look at the right-hand column on this particular slide, you'll see that there were, in fact, I think, 7 patients who preferred placebo on that basis, leaving 16 patients who preferred amitriptyline. And if I look at this particular analysis here, it's the 16 successes out of 23. The proportion is rather similar to what I had before, but of course, the evidence is less because I'm no longer regarding cycles as being independent. Now I'm only regarding averages per patient as being independent, and the exact probability is 0.093. So the result is no longer significant. It's not even perhaps what some optimistic medic might call a trend towards significance. So the 95% confidence interval proportion is 0.4708. It crosses 0 to 0.8679. So the question is, can we do better? These tests are, of course, very limited. The first makes a strong assumption of independence that might be reasonable for a limited purpose of testing a strict null hypothesis. If it is the case that amitriptyline is no better than placebo for any patient, then by hypothesis, these particular results are independent, because there can be no random patient by treatment interaction if all of the patients, if amitriptyline works for no patient. So it's a strict null hypothesis. It might be worth testing in the first round of tests, but of course it has a limited interest. The second, however, throws away much information. Neither of them enables us to say anything about personal response. So this is where we need a mixed model. And one approach is a logit binomial mixture, which is very common in applying random effects to binary outcomes. And we're now going to illustrate this. So we're going to assume that we have a logistic type response. YIR is little YIR is an indicator value recording whether amitriptyline or placebo is preferred in a particular cycle, I for the patient, R for the cycle for that patient, and it can take on the value 0 or 1. We now imagine that we're going to model how this particular probability theta I sub r varies on the logit scale, and so we have the log odds ratio, or the logit rather, which I'm going to set equal to eta IR, and then eta itself is assumed to have some particular distribution here, a normal distribution, has an overall effect capital tau, and a random deviation tau sub I, and tau sub I I'm going to assume is distributed normal naught psi squared. I just remind you, YIR is a binary outcome indicating a preference for A if equal to 0 or for B if equal to 1. This model is an implementation of the common binomial logit mixture, which was used, for example, by Farquhar Etzet and John Whitehead in an important paper in Statistics in Medicine of 1992, or actually this may be in Applied Statistics. There were two papers that they had. I'm not quite sure which one I'm citing here. I can analyze this using Procadel mix of SAS. This is a particular routine, very fine code developed by Russ Wolfinger for SAS, and this particular is just a particular implementation of that particular model. I'm not going to have a look at this in detail. These are the parameter estimates. Tau is an estimate on the logit scale. This is its standard error, and this is the confidence interval for it here. The result is just significant using conventional standards of significance. I can actually have the same result on the probability scale if I want to, and this is 0.6837. I'm not quite sure what's going on here, but obviously SAS doesn't particularly like telling me things about the t-value and so forth on this particular scale. But I've just used this as a way of getting the estimate of the probability which corresponds to that particular logit. This shows that there's strong shrinkage in this particular example. This particular example, unlike the example we've been having a look at so far, which was an example simulated for the purpose of illustrating the various points, this particular example is actually genuine. Here I have jittered the points to enable you to see the differences, otherwise they'd just be on top of each other. And you can see that some fairly strong shrinkage has occurred, but that there is a little bit of variation above and beyond that which will be predicted by pure binomial variation. And for that reason, the points are not all horizontally along the average value here, the overall average value. This is the same thing in GenStat, but using the HGLM approach of Lee and Nelder. Here I've also plotted the points on the line of equality, so you can see them. And this is the this is the shrinkage line. You can see that the estimates are fairly similar to what we had before, but the system is slightly different. What about random effects for genuine binary outcomes? That was an analysis of preferences. Here I'm going to take a case where we actually have binary outcomes at the level of the episode within a cycle. So it's not a question of taking a preference and declaring that preference to be binary, either B or A. The actual observation itself is binary. And I'm going to use a very strange example for these crossover trials in infertility. If you have a look at my book on crossover trials, a second edition of which I think is 2002, and you'll see that I describe crossover trials, infertility, as being an indication in which crossover trials are unsuitable. And my argument at the time was, well, it's not a reversible condition. Basically, you're not going to take a particular couple and say, well, now that you have achieved conception, then we're not going to try and rerun time and see whether you conceive it again with another treatment. Essentially, the situation is unreversible. That's a success, and they would not proceed to the second period of treatment if they were if the successful conception was achieved in the first cycle. However, sometime after the second edition appeared, I began to worry about this. I began to think, well, this is all very well, Stephen, but why are you being so negative? You're looking at this as a glass half empty. Think of it this way. You run a parallel group trial. Patients have either been offered treatment A or treatment B. And then if the treatment is unsuccessful, they're simply offered the second treatment. That means you've got some extra data. How can having more data be worse than having fewer data? So there has to be a way in which these can be analyzed. And I then actually realized that when you start to think about it this way, you see that the data are in fact missing at random, because you know why they're missing. They're missing because conception has been achieved. So from that point of view, you can deal with them. And this is the project that Volkanyo Makabati worked on. So here are two views about crossover trials in infertility. The effective treatment is not reversible. There'll be missing data. So the indication is inherently unsuitable for crossover trials. That was the wrong opinion. I now believe it to be a wrong opinion that I once held. Pro is what we have as a parallel group trial with some extra data. How can more data be worse than fewer? There must be a way of analyzing this. That's what I now believe. So I'm going to have a look at an example here. This is from Cohen et al. In 1998. 74 couples were treated for male infertility. The treatments were intrauterine insemination alone, N for natural, or including ovarian hyperstimulation, S for stimulated. And treatment was in up to 6 cycles, or a total of 320 cycles, and a mean of 4.3 per couple. And you'll see this particular example discussed some years ago in Statistics in Medicine. So we can use the random effects model of Etzet and Whitehead again. Here we have it actually at the level, however, of the individual cycle. Before it was at the level of the individual episode, sorry, before it was at the level of the preference for the cycle. So here is a summary of the data from Cohen et al. Essentially, what you have is you have the number of patients in total on the 2 cycles. There were 36 on the second sequence, or 2 sequences, sorry, 38 on the first sequences. And you've also recorded whether they have achieved pregnancy or not. And 21 didn't. They went right through the particular series of 6 cycles without pregnancy being achieved. but 17 did. And essentially what one records here is whether an event occurs or not. Then, of course, the couple did not go on to receive the following data. So of the 17, for example, who were given the cycle NSNS, then 2 achieved pregnancy or conception on episode 3. And that means that they were using the non-stimulated form. There are the 2 here, 12 on and the final sixth occasion. That is, in fact, when they were stimulated here. So this is a sort of condensed summary of the results. You can use these if you want. By the way, these data are available on the web somewhere. I think I give a reference. So now here we've carried out 2 analyses. We first of all carried out a null analysis in which we don't use the treatment effect, and then we've only fit the period effect, and of course the subject effect is fitted. And then a second one in which we use period versus treatment. By the way, note that really one hasn't used much in the way of randomization here. Patients have been randomized to 2 sequences, but the sequences are essentially always of the form that you switch the treatment. You're given N and then S and N and then S and then an S, or S and then N, etc. So it's not the same sort of design that we used before. In my opinion, this is perhaps a weakness of this particular approach. I'm not quite sure why they didn't randomize pairs of cycles, but anyway, they didn't. So we have here the result without period, and then the result, with period without treatment, and then the result fitting treatment. And I'm comparing 4 particular approaches. 2 of them ought to be essentially the same. They are SAS and R. They're simply, as far as I'm aware, the local implementations in SAS and R of the particular model that I've outlined. And you can see that the treatment estimate is the same here, 0.64 for the 2, a slightly different standard error. The intercept, for some reason, the standard error is bafflingly different in SAS and R. I've no idea why. I did think about this at one time, but I've completely forgotten what particular conclusion I came to. And I just hold this up as a warning that you can find surprisingly different answers when you use different software. A particularly notorious case, by the way, is one of when Reinhard Bergman and colleagues some years ago had a look at the Wilcoxon test applied by, I think, 16 different packages to one particular problem, and they had 12 different answers amongst the 16 different packages. This was to do with whether continuity corrections were used or not, at what point an asymptotic result was applied, yes or no, how exactly ties were handled, and so forth. So all of these particular details could make a difference. So it's just a warning here. Winbugs, of course, it's a Bayesian approach. The result is slightly different anyway. But you can see that the treatment estimate is, well, it's in the same ballpark, and the standard error also. The GenStat approach is a GenStat approach using the Lee-Nelder hierarchical generalized linear model, which deals with the random effect itself rather differently. And you can see, again, same sort of ballpark, standard error rather similar to those for the others, but there are nevertheless some differences. So this is my second example. This is of infertility. This is Gregorio et al. This is male infertility. And they compared intrauterine insemination or timed intercourse. And the 62 couples were divided at random into 2 equal groups, either AU, in which they received either IUI or TI for 3 cycles. If failed to achieve pregnancy, switched to the other treatment. So here it was 3 menstrual cycles. It's a fortunate use of the same word, if you like, for which it was an attempt to achieve pregnancy. And then they were switched to the other treatment for the remaining 3. So what do we observe? If you regard this as being an ABA crossover, and you code the outcome as 0 for pregnancy and 1 for pregnancy, then what you will observe is the following. You will observe the frequency of the following outcome: 0, 0, which means that pregnancy was not achieved on either treatment; 0, 1, which means pregnancy was not achieved on the first treatment, but it was on the second; and 1. 1 essentially, if it occurs in the first set of treatment, the first treatment that's given, leads to the trial being discontinued. If conception is achieved, then in that case, it's impossible to carry on with the trial. So that 1 represents a marginal collapse of the possible sequence of results, the theoretically possible sequence of results 1, 0 and 1, 1. The occurrence of 1 in the first period suppresses the result in the second. So we can have a look at the results as follows. If we look at the TI/IUI sequence, then there were 20 couples who achieved conception under neither of the 2 treatments. There were 7 who did not under TI, that was timed intercourse, but who did under the artificial insemination. And there were 4 couples who achieved pregnancy, conception under the first treatment, but not the second. And then you have the same thing for the other alternative sequence of treatment. So this is what you're going to observe. Now, how could we analyze this? Well, first of all, we could say, well, let's just ignore period 2 for the moment. Let's just have a look at period 1 and see what we can say about that. Well, then in that case, we're comparing TI to IUI in period 1, and we have a standard 2-by-2 table, and we can analyze this. And the highlighted here, what's been highlighted in red and purple, are the results essentially from period 1. That's what we get. Then what we can do is we could also do the same for period 2, but now we're doing a conditional analysis. Now we're only analyzing those particular couples who failed to achieve pregnancy in the first course of treatment, and we're having a look at what happened in the second course of treatment, and we're doing that. So we treat the marginal table using the first period data and the conditional table using the second period data as 2 strata. This is effectively what happens in a Mantel-Haenszel test, and it's effectively what happens in the log rank test also. It's a standard device of conditioning of what's happened so far when looking at survival data. Because the second table is a conditional table, it can be treated as if it was independent of the first, and we apply the Mantel-Haenszel procedure using period as a stratifying factor. So this is an alternative tabulation of the data. Period 1 was pregnancy achieved, no, yes with the treatment, and period 2, no, yes with the treatment. And we then do the Mantel-Haenszel test, and we can calculate the log odds ratio associated with the Mantel-Haenszel procedure, and we can calculate the Robbins, Breslow, Greenland standard error as well associated with that. So all the standard things that we do when carrying out this sort of analysis, we simply recognize that the data can be arranged in this particular way. I'm baffled as to why it took me so long to see this. Here, by the way, are some programs to accompany the Macabati and Sen paper. If you have a look, there's the URL for the particular page is given at the bottom of this particular slide, and these are some particular analyses in various particular packages. I haven't looked at them for a while. I hope they still all work. Right. So that was binary data, which leaves us about a quarter of an hour to have a look at planning. So just to warn you that exactly as with crossover trials, carryover is potentially a problem for N-of-1 trials. It's possible that because one has many cycles, one's ability to model carryover is greater than with standard ABBA crossover trials. But I myself am very skeptical as to whether one can make progress this way. Basically, I think that carryover and dealing with it is a matter of biology and medicine. It's not a matter of statistics. And a lot of the modeling of carryover in crossover trials, I characterize, perhaps unkindly, as being some sort of a dream in which mathematics triumphs over biology. Basically, I think it's a question of the subject matter specialists, and the statisticians are misleading them if they think they have some magic way of dealing with it. You have to think hard about what the reasonable effect is in carryover and whether or not the particular indication is therefore suitable for N-of-1 trials or crossover trials. I'm not going to go over this in detail here. So just to say, the key to controlling for carryover is adequate washout, but there are two ways you can do this. You can actually wash out in the sense of not giving the patient at all treatment in a particular period. Clearly, this has ethical problems, practical problems. The other is to use what I call an active washout period. That is simply to recognize that all you have to do is treat the patient long enough for the effect of the previous treatment to be negligible. So you restrict measurement towards the latter part of the observation period for a particular episode of treatment. It's assumed that the effect of the previous drug will have been eliminated, and also that the effect of the current drug will have reached steady state. We've not modeled period effects, but we've modeled cycle effects. In general, for within patient design, trial period effects may be expected to be not well defined. Patients are not recruited simultaneously. Thus, something we should not forget is that period 2 for one patient may be before period 1 for another. So periods from the patient point of view imperfectly reflect temporal processes. They could reflect some secular improvement or deterioration applicable to all, but they could also reflect other things that are happening in the environment. For example, you might be looking at hay fever or asthma, let's say, rather, and it might well be that for a particular patient, you're studying them during the hay fever season, not for other patients, and so forth. So there are clearly trend effects here, which may not be necessarily global. And cycle effects are a way of influencing local trend effects, and this may be preferable. This is one reason why I think the analysis in terms of paired episodes of treatments, episodes in pairs, which we've referred to as cycles, in which the patient is given A followed by B or B followed by A, why this is a way of dealing with local trend effects. But there's another important effect of time. Because patients are recruited over time, we shall know more about patients recruited earlier than those recruited later. So I'm picking up on something I said at the end of the last lecture. If we want to produce shrunk estimates for patients, we could do better by revisiting the predictions we made for earlier patients once we have more patients. But that represents something of a logistic challenge. I'm not going to address this problem here, but I'm just raising it again as an issue which is worth thinking about for those of you that are actually doing this for real, rather than, as is the case for me, pontificating about it. Calculating sample sizes for N-of-1 trials may be complicated because for certain purposes, more than one component of variation may be involved. This means if on which to have exact power calculations would be a distributional issue, since a simple t-distribution does not apply. And the issue is how to handle the non-centrality parameter, and it becomes complex. In practice, I think asymptotic theory should suffice. But wherever an analysis is equivalent to a single summary measure, then exact calculation, I put this in brackets because, in inverted commas, because we shouldn't forget that the major uncertainty with sample size calculation, and this applies to all clinical trials, is essentially the assumed parameter values we put in. The basic idea of sample size calculation, which I'm sure many of you are familiar with, is the following. The type 1 and type 2 error rates we tolerate determine what might be called decision precision. For example, when looking at continuously distributed outcomes and then therefore employing the normal distribution, we might have something like omega equals Z alpha over alpha. I've put the 2 there because we're going to usually be using two-sided tests, plus Z beta. Beta and alpha are our type 1 and type 2 error rates, and this is the target non-centrality. We want to make sure that we end up with a standardized result which is equal to this. The beta governs the power that we're going to have, and the alpha is the type 1 error. On the other hand, if we look at the signal that the trial will deliver, the standardized signal, then that's going to be in terms of some clinically relevant difference, delta, of the numerator. And then the denominator will involve some expression for the variance. I've put here V as a function of all of those various particular components. Psi squared is the treatment effect variance from patient to patient. Sigma squared is the within patient, within cycle variance. N is the number of patients. K is the number of cycles. So these together will determine what our standard error is. That standard error will be divided into delta. That will be our standardized difference. And that is what we need to make equal to Z alpha over 2 plus Z beta. And then essentially we solve for the particular things we're trying to fix here. Note, by the way, a point about delta. I like to think of delta as the difference we would not like to miss. It's not a difference you would expect. We're carrying out a trial in order to try and discover things. Let's not have expectations really as part of it. It's essentially like a screening procedure. We want to make sure we don't miss treatments with a particular value. The situation for N-of-1 trials is more complicated because there are two things we can vary. We can vary N and K, and their effect on the particular standard error is complex. But otherwise, basically, it's the same sort of thing. What we're trying to do is we're fiddling around with the values of N and K, given values of psi squared, sigma squared, alpha, and beta that we've assumed, and we're seeing whether they deliver the results. And here is a reminder of some variance formulae, and this shows why there are different particular calculations we could engage in. We could be looking for different types of things, and depending on what we're looking for, then in that case, we should get different answers. I have a paper in, I'm not quite sure where it is, maybe Pharmaceutical Statistics or possibly Statistical Methods in Medical Research, I can no longer remember, which actually looks at this sample size determination and gives some advice, which I hope is correct. So I'm going to skip over this. So you can have a look at this yourself if you like. This is some code, by the way, in SAS. And some results. And I think what I'm doing there is I'm using the sort of random effect approach. This is using the fixed effect approach, which actually turns out bafflingly to be rather more complicated for reasons I won't explain. And this is a particular diagram using the same sort of thing in GenStat. And here I'm looking at a sample size calculation for N-of-1 trials. And essentially, I'm looking at different purposes here. The random effect one is when I'm trying to say something about patients in general, perhaps including those outside the trial. The fixed effect one, where my particular ambition is more limited, and I'm simply trying to say something about the patients in the trial. And not surprisingly, you can see that the sample size, the number of patients I need to study, in both cases, reduces as the number of cycles increases. But in all cases, essentially, I have more power, and so I can get by with fewer patients for the fixed effects approach rather than for the random effect approach. I also have formula, again, I don't really have time to go into this, for the number of cycles to study a future patient, if for a particular purpose, if you want to be able to guarantee for a particular patient, or not guarantee, but to predict some sort of probability of success long-term with a treatment, then you might want to do this. And here are some standard errors for two particular approaches, or three, in fact. I've got the standard error using previous data only, which I've fixed to be equal to 1, just to illustrate here. And that's the psi squared variance essentially is relevant here. I'm imagining I've studied a huge number of patients, and I'm just predicting for you what your result will be if you stay on this treatment for a long time using their particular results. That's the naive standard error that would apply using previous data only. Sorry, this is the standard error using previous data only. The naive one is just using your results, and the shrunk one is using both. And you can see that here for this particular example, but everything depends on the parameter values. Here, the within patient variability is rather high. Here, it actually takes quite a large number of cycles studied for you before using your data alone would be better than using previous data only. But obviously, we want to use neither of these two methods. What we want to be using is the shrunk approach. Further issues, the previous theory is on the assumption that the nuisance parameters themselves are reasonably well estimated. This implies reasonable number of degrees of freedom for estimating the two variance components, a reasonable number of patients for estimating the overall mean, but further discussion of these issues is beyond the coverage of this course. So that's it, the course is over, but the last bullet point is a lie. There isn't really much time for your questions, but I'm very happy to answer any questions I can in what time remains, and I can certainly hang on for a quarter of an hour if that's useful for people. Otherwise, if you send your questions to Michael, I think, then I will try and answer them by email in due course. Thank you.
Thank you so much, Stephen. Very challenging in some respects and enlightening couple of talks today. Look, we have a few minutes for questions, and thank you so much for agreeing to stay on for a bit. So please, anyone, put questions you have in chat. Could I just ask just a sort of a general perspective question, Stephen, about N-of-1 trials, I suppose, apart from crossover trials, they're really the only trial design where you can actually observe individual treatment effects and treatment effect heterogeneity across individuals. What's your perspective, I guess, from practical experience on when there is substantial treatment effect heterogeneity and therefore the potential for personalized medicine? Because if there's no treatment effect heterogeneity, then there's no reason to go hunting for individual subject characteristics that would be sort of prognostic of sort of higher or lower treatment effects. Do you have any sort of thoughts on that sort of issue?
Yeah, I think unfortunately, in a lot of fields, people have tried to shortcut the process of judging individual effects by carrying out a so-called responder analysis. And what they've done is they have categorized patients as being responder, non-responder, based on criteria that are often really pretty arbitrary. And in doing this, they've ignored the fact that there's a great deal of chance involved in this. And so they've tended to assume that if a particular trial has got that 70% of the patients under the better treatment were responders, that therefore the treatment works in 70% of the patients and not in 30%. And this is completely unjustified. And I think the main shame about N-of-1 trials is it's obvious that we can't carry them out in many indications, but it's a bit of a disgrace that we haven't carried out more. If we carried out more, they would have taught us to think about response properly. And we would have learned practically from examples, and examples are always telling, to what extent personal response is important, more important than simply with in-person variability. And we haven't looked at that. And so I think that the whole edifice of personalized medicine is based on a premise that we haven't really examined properly. We haven't looked to see what the scope is for individual response. We assume it's there, but we don't know it's there. And I think that's something of a disgrace. And that's one reason why I'm quite enthusiastic about doing more in the way of N-of-1 trials, because we will gradually get more examples. Of course, the examples from one disease don't necessarily carry over to another, but it will help us to think about things in this way. And I think we should be doing that more. There have been some successes for personalized medicine, of course. But also, I think that the field has been one of very expensive failure over the last 30 years or more or less, since we were promised that clinical trials were going to get smaller and quicker, cheaper, and that hasn't happened. The reverse has happened. So I think there is a big problem here.
What are the major barriers? Other point. Oh, sorry, go ahead.
Yeah. I mean, to make one other point, I mean, I think also the pharmaceutical industry lost its ambition. If you take, if I take an example of one particular disease, which I think in my own lifetime was completely revolutionized its treatment, and that was gastric ulcers. Part of the story, of course, was to do with Australia, where the role of H. pylori bacteria in infection was proved. But even before that, we had the development of the H2 antagonists and simultaneous proton pump inhibitors. And then once the H. pylori role was established, we had antibiotics to go with it. And then we also had a breath test that could check whether you were needed treatment that way or another. But basically what happened there was that more or less gastric ulcers to a large extent were wiped out. Before that, you were sent to the surgeon. And that's a huge success. And yes, there's a certain degree of personalization in it, obviously testing for H. pylori presence, but even if you didn't have H. pylori, then the powerful antacids were really very, very effective. And empirical treatment was effective. And instead, what we then embarked on was developing expensive medicines for almost nobody. It strikes me a really lack of ambition. We were somehow blinded by the science.
That sounds like a good note to conclude on here, that let's not get blinded by the science.
Sorry, I don't want to.
Could you please join me in virtually thanking Stephen, not only for today, but for the presentations 2 weeks ago. And as he said, the slides will be made available. And so there's been a lot to contemplate over these last 2 sessions, but I'm sure we'll be all the better from studying them closely. So again, I wanted to thank you very much, Stephen, and thank you to the audience for coming along.
Okay, thanks for the invitation and thanks for your attention.